Hi students! Warm welcome to today's session. In this session we are going to see about antioxidants, anti-caking and anti-foaming agent. Okay, let's get into today's session. Antioxidants are molecules that inhibit or quench free radical reactions and delay or inhibit cellular damage. Though the antioxidant defenses are different from species to species, the presence of the antioxidant defense is universal. Antioxidants exists both in enzymatic and non-enzymatic forms in the intracellular and extracellular environment. Normal biochemical reactions, increased exposure to the environment, and higher levels of dietary xenobiotics, result in the generation of reactive oxygen species, ROS, and reactive nitrogen species, RNS. ROS and RNS are responsible for the oxidative stress in different pathophysiological conditions. Cellular constituents of our body are altered in oxidative stress conditions, resulting in various disease states. The oxidative stress can be effectively neutralized by enhancing cellular defenses in the form of antioxidants. Certain compounds act as in vivo antioxidants by raising the levels of endogenous antioxidant defenses. Expression of genes encoding the enzymes such as superoxide dismutase, SOD, catalase, CAT. Glutathione peroxidase, GSHPX, increases the level of endogenous antioxidants. Antioxidants can be categorized in multiple ways. Based on their activity, they can be categorized as enzymatic and non-enzymatic antioxidants. Based on their solubility in the water or lipids, the antioxidants can be categorized as water-soluble and lipid-soluble antioxidants. The antioxidants can also be categorized according to their size, the small molecule antioxidants and large molecule antioxidants. Let's see the classification one by one. First classification: enzymatic and non-enzymatic antioxidants. Enzymatic antioxidants work by breaking down and removing free radicals. The antioxidant enzymes convert dangerous oxidative products to hydrogen peroxide, H2O2, and then to water, in a multi-step process, in presence of cofactors such as copper, zinc, manganese, and iron. Non-enzymatic antioxidants work by interrupting free radical chain reactions. Few examples of the non-enzymatic antioxidants are vitamin C, vitamin E, plant polyphenol, carotenoids, and glutathione. Second classification is based on their solubility in the water or lipids. The antioxidants can be categorized as water-soluble and lipid-soluble antioxidants. The water-soluble antioxidants, example vitamin C, are present in the cellular fluids such as cytosol, or cytoplasmic matrix. The lipid-soluble antioxidants, example vitamin E, carotenoids, and lipoic acid, are predominantly located in cell membranes. Third one is according to their size, the small molecule antioxidants and large molecule antioxidants. The small molecule antioxidants neutralize the ROS in a process called radical scavenging and carry them away. The main antioxidants in this category are vitamin C, vitamin E, carotenoids, and glutathione, GSH. The large molecule antioxidants are enzymes, SOD, CAT, and GSHPX, and sacrificial proteins, albumin, that absorb ROS and prevent them from attacking other essential proteins. To understand the mechanism of action of antioxidants, it is necessary to understand the generation of free radicals and their damaging reactions. First we will see about generation of free radicals. The generation of ROS such as O2, singlet oxygen, O2 monos, superoxide anion radical, star OH, hydroxyl radical, RO star, alkyl radical, ROO star, peroxyl radical, H2O2, hydrogen peroxide, LOOH, lipid hydroperoxide. Begins with rapid uptake of oxygen, activation of NADPH oxidase, and the production of the superoxide anion radical. These ROS can act by either of the two oxygen-dependent mechanisms, resulting in the destruction of the microorganism or other foreign matter. The reactive species can also be generated by the myeloperoxidase halide H2O2 system. The enzyme myeloperoxidase, MPO, is present in the neutrophil cytoplasmic granules. In presence of the chloride ion, which is ubiquitous, H2O2 is converted to hypochlorous, HOCl, a potent oxidant and antimicrobial agent. ROS are also generated from O2 and H2O2 through respiratory burst by Fenton or Haber-Weiss reactions. The enzyme nitric oxide synthase produce reactive nitrogen species, RNS, such as nitric oxide, NO*, from arginine. An inducible nitric oxide synthase, INOS, is capable of continuously producing large amount of NO*, which act as a O2- quencher. The NO* and O2- react together to produce peroxynitrite, ONOO-, a very strong oxidant, hence, each can modulate the effects of other. Although neither NO* nor O2- is a strong oxidant, peroxynitrite is a potent and versatile oxidant that can attack a wide range of biological targets. Peroxynitrite reacts with the aromatic amino acid residues in the enzyme, resulting in the nitration of the aromatic amino acids. Such a change in the amino acid residue can result in the enzyme inactivation. However, nitric oxide is an important cytotoxic ether molecule in the defense against tumor cells, various protozoa, fungi, helminths, and mycobacteria. The other sources of free radical reactions are cyclooxygenation, lipooxygenation, lipid peroxidation, metabolism of xenobiotics, and ultraviolet radiations. Damaging reaction of free radicals. ROS-induced oxidative stress is associated with the chronic diseases such as cancer, coronary heart disease, CHD, and osteoporosis. Free radicals attack all major classes of biomolecules, mainly the polyunsaturated fatty acids, PUFA, of cell membranes. The oxidative damage of PUFA, known as lipid peroxidation, is particularly destructive, because it proceeds as a self-perpetuating chain reaction. Let's see about lipid peroxidation reaction. In this reaction, LH is the target PUFA and R* is the initializing, oxidizing radical. Oxidation of the PUFA generates a fatty acid radical, L*, which rapidly adds oxygen to form a fatty acid peroxyl radical, LOO*. The peroxyl radicals are the carriers of the chain reactions. The peroxyl radicals can further oxidize PUFA molecules and initiate new chain reactions, producing lipid hydroperoxides, LOOH, that can break down to yet more radical species. Lipid hydroperoxides always break down to aldehydes. Many of these aldehydes are biologically active compounds, which can dias from the original site of attack and spread the attack to the other parts of the cell. Lipid peroxidation has been widely associated with the tissue injuries and diseases. Oxygen metabolism generates OH, O2*, and the non-radical H2O2. The OH is highly reactive and reacts with biological molecules such as DNA, proteins, and lipids, which results in the chemical modifications of these molecules. There are several research reports on the oxidative damage of DNA due to the OH. The OH reacts with the base pairs of DNA, resulting in the oxidative damage of the heterocyclic moiety and the sugar moiety in the oligonucleotides by a variety of mechanisms. This type of oxidative damage to DNA is highly correlated to the physiological conditions such as mutagenesis, carcinogenesis, and aging. The addition reactions yield OH adduct radicals of DNA bases, whereas the allyl radical of thymine and carbon-centered sugar radicals are formed from the abstraction reactions. As shown in the figure, the OH* reacts with the guanine of the DNA to produce the C8-hydroxy adduct radical of guanine, which is converted to the 2,6-diamino-4-hydroxy 5 form. Amidopyrimidine upon reduction and ring opening reactions. However, the C8-hydroxy adduct radical of guanine is converted to the 8-hydroxy guanine upon oxidation reaction. The OH* radical reacts with the heterocyclic moiety of the thymine and cytosine at C5 and C6 positions, resulting in the C5O and C6O adduct radicals, respectively. The oxidation reaction of these adduct radicals with water, followed by deprotonation. Results in the formation of the cytosine glycol and thymine glycol, respectively. Overall, the reactions of the OH star with the DNA bases result in the impaired double-stranded DNA. As shown in the figure, the OH star reacts with the sugar moiety of DNA by abstracting an H atom from C5 carbon atom. One unique reaction of the C50-centered radical of the sugar moiety in DNA is the addition to the C8 position of the purine ring in the same nucleoside, example guanine. This intramolecular cyclization results in the formation of the 8,5-cyclopurine 2-deoxynucleosides. The reactions of carbon-centered sugar radicals result in the DNA strand breaks and base-free sites by a variety of mechanisms. Proteins are oxidatively damaged by the combined action of activated oxygen species and the trace metal ions such as Fe2+ and Cu2+. The amino acids lysine, proline, histidine, and arginine have been found to be the most sensitive to oxidative damage. Recent studies indicate that a wide range of residue modifications can occur, including formation of peroxides and carbonyls. Generation of the carbonyl residue is a useful measure of oxidative damage to proteins. Thus, the oxidative damage to tissue results in the increased amount of oxidized protein. Low levels of antioxidants have been associated with the heart disease and cancer. Antioxidants provide protection against a number of disease processes such as aging, allergies, analgesia, arthritis, asthma, atherosclerosis, autoimmune diseases, bronchopulmonary dyspepsia, cancer. The other disorders to which antioxidants provide protection are cataract, cerebral ischemia, diabetes mellitus, eczema, gastrointestinal, inflammatory diseases, genetic disorders. Following section elaborates the mechanism of action of the radical scavenging activities of various natural antioxidant molecules. Modulation of free radicals by natural antioxidants. Two types of antioxidants, namely the enzymatic antioxidants and non-enzymatic antioxidants, modulate the free radical reactions. Body protects itself from ROS by using enzymatic antioxidant mechanisms. The antioxidant enzymes reduce the levels of lipid hydroperoxide and H2O2, thus they are important in the prevention of lipid peroxidation and maintaining the structure and function of cell membranes. Examples of the enzymatic antioxidant are CAT, GSHPX, SOD, and peroxyredoxin 1,4. SOD, located in the cytosol and mitochondria, catalytically convert the O2 star into oxygen and H2O2 in presence of the metal ion cofactors such as copper, Cu, zinc, Zn, or manganese, Mn. The enzyme CAT, present in the peroxisome, converts H2O2 to water and oxygen. GSHPX are found both in the cytoplasm and extracellularly in almost every human tissue. GSHPX convert the H2O2 into the water. The enzyme GSHPX has strong activity towards both H2O2 and fatty acid hydroperoxides. The enzyme peroxyredoxin catalyzes the reduction of H2O2, organic hydroperoxides, and the peroxynitrite ONOO. The different expression profiles, subcellular locations, and substrates of the antioxidant enzymes reveal the complex nature of the ROS biology. Clearly, the antioxidant enzymes play a major role in the prevention of oxidative damage. CAT, GSHPX, and SOD show synergistic effect in the scavenging of O2 star. Let's see about natural antioxidants. First one is vitamin E. Vitamin E, alpha-tocopherol, is an ancient lipid-soluble antioxidant that functions as a chain breaker during lipid peroxidation in cell membranes and various lipid particles, including low-density lipoprotein, LDL. It functions to intercept lipid peroxyl radicals, LOO*, and to terminate the lipid peroxidation chain reactions. The resultant tocopheroxyl radical is relatively stable and in normal circumstances insufficiently reactive to initiate lipid peroxidation itself, which is an essential criterion of a good antioxidant. It should be noted that Vitamin E exerts antioxidant effects by scavenging lipid peroxyl radicals in vivo as well as in vitro systems. However, vitamin E is not an efficient scavenger of OH* and alkyl radicals, OR*, in vivo. Vitamin C or ascorbic acid too, is a water-soluble free radical scavenger. Moreover, it regenerates vitamin E in cell membranes in combination with GSH or compounds capable of donating reducing equivalents. Vitamin C changes to the ascorbate radical by donating an electron to the lipid radical in order to terminate the lipid peroxidation chain reaction. The pairs of ascorbate radicals react rapidly to produce one molecule of ascorbate and one molecule of dihydroascorbate. The dihydroascorbate does not have any antioxidant capacity. Hence, dihydroascorbate is converted back into the ascorbate by the addition of two electrons. The last stage of the addition of two electrons to the dihydroascorbate has been proposed to be carried out by oxidoreductase. Antioxidant potential of vitamin A3 was first described by Monahan and Schmidt, who reported that vitamin A can protect lipids against rancidity. Several reviews have appeared to outline the basic structural and metabolic characteristics of vitamin A and information about its potential as antioxidants in relation to the heart diseases. Vitamin A has a vital antioxidant contribution in protecting human LDL against copper-stimulated oxidation. Next natural antioxidant is bioflavonoids. Bioflavonoids are a group of natural benzo-G-pyrene derivatives and are found to possess strong antioxidant activities. Bioflavonoids widely distributed in fruits and vegetables, are reported to exert multiple biological effects including free radical scavenging activity. It has been reported that the bioflavonoids have a protective effect on the DNA damage induced by the hydroxyl radicals. One of the mechanisms that explains the protective effect of the flavonoids on the DNA is the involvement of the chelating metal ions, such as copper or iron. The flavonoids complexed with the copper or iron prevent the generation of the ROS. There are different examples of bioflavonoids. First one is quercetin. It is a flavonol, known to protect DNA from oxidative damage resulting from the attack of OH*, H2O2, and O2* on the DNA oligonucleotides. On the contrary, quercetin is also reported to be carcinogenic agent. According to the reports, quercetin has opposite effects on DNA damage induced by cupric ion depending on the concentration of cupric ion. At the low concentration of cupric ions, that is less than 25 micromolar, quercetin exhibit a protective role. While, at higher concentration of cupric ion, that is greater than 25 micromolar, quercetin enhances the damage to DNA by ROS. Therefore, it is very important to consider the concentration of the chelating metal ions, such as copper or iron, while evaluating the protective or degenerative effects of quercetin and other bioflavonoids. Next example is anthocyanidine. It is a class of flavonoid, are potential antioxidants and their effectiveness in the inhibition of the lipid oxidation is related to their metal ion chelating activity and free radical scavenging activity. Three structural groups are important determinants of the radical scavenging activity of anthocyanidins. First, the orthodihydroxy structure in the B ring. Second, the 2,3 double bond in conjugation. Third, the 4-oxo function in the C ring. Flavonoids form complexes with the metal ions by using the 3 or 5 hydroxyl and 4 keto substituents or hydroxyl groups in ortho position in the B ring. Another natural antioxidant is carotenoids. They are among the most common lipid-soluble phytonutrients. Lycopene and beta-carotene are the prominent carotenoids among other 600 different compounds. The biosynthetic pathway as shown in figure, demonstrates the synthesis of carotenoids from phytoene, which is synthesized from 2 molecules of geranyl pyrophosphate. Carotenoids are well known to scavenge the peroxyl radicals more efficiently as compared to any other ROS. The peroxyl radicals generated in the process of lipid peroxidation can damage the lipids in the cell wall. Scavenging of peroxyl radicals can disrupt the reaction sequence and prevent the damage to cellular lipids. The long unsaturated alkyl chains in carotenoids make them highly lipophilic. Carotenoids are known to play an important role in the protection of cellular membranes and lipoproteins against the ROS due to their peroxyl radical. Examples of carotenoids. First example is lycopene. It is the most potent antioxidant naturally present in many fruits and vegetables. The high number of conjugated double bonds in lycopene endows it the singlet oxygen quenching ability. Lycopene demonstrate the strong singlet oxygen quenching ability as compared to the alpha tocopherol or beta-carotene. Next example is beta-carotene. It is a naturally occurring orange-colored carotenoid, abundantly found in the yellow-orange fruits and in dark green leafy vegetables. Beta-carotene demonstrates potential antioxidant property due to its chemical structure and the interaction with biological membranes. It is well known that the beta-carotene quenches singlet oxygen with higher efficiency as compared to the alpha tocopherol. In addition, it is also known that the Z isomers of the beta-carotene possess antioxidant activity in vitro. Furthermore, the beta-carotene can be converted into the two molecules of vitamin A by the beta-carotene deoxygenase catalyzed cleavage. Next natural antioxidant is hydroxycinnamates. The examples are ferulic acid, caffeic acid, cynapic acid, para-coumaric acid. It is widely accepted that the dietary antioxidants that protect LDL from oxidation can prevent the atherosclerosis and coronary heart disease. Hydroxycinnamic acids and their conjugates prevent oxidative damage to the LDL. The in vitro studies involving human LDL as the oxidizing substrate showed that the hydroxycinnamic acids have higher antioxidant activity as compared to the corresponding hydroxybenzoic acids. The antioxidant activity of the derivatives of the hydroxycinnamatides is clearly correlated with the hydroxylation and methylation patterns of the aromatic ring. The antioxidant efficiency of the free hydroxycinnamatides on the human LDL oxidation in vitro decreases in the order of caffeic acid, cynapic acid, ferulic acid, para-coumaric acid. The presence of the ortho-dihydroxy group in the phenolic ring, as in caffeic acid, enhances the antioxidant activity of hydroxycinnamic acids toward human LDL oxidation in vitro. The radical scavenging antioxidant mechanism of the hydroxycinnamic acids are similar to that of the flavonoids. Because of their ability to donate an hydroxyl hydrogen and resonance stabilization of the resulting antioxidant radicals. The ortho-dihydroxy substituents also allow the metal ion chelation similar to that of flavonoids. Let's see about some other natural antioxidants. First one is theaflavin, and theaflavin 3-gallate possesses in vitro antioxidative properties against lipid peroxidation in the erythrocyte membranes and microsomes. They also suppress the mutagenic EECs induced by H2O2. Theaflavins inhibit the H2O2-induced cleavage and mutagenicity of the DNA single strand. In general, theaflavins scavenge the free radicals to produce antioxidative and anti-mutagenic EECs. Apart from the aromatic hydroxyl groups of theaflavins, the gallic acid moiety is essential for their antioxidant activity. The theaflavin 3-gallate is a stronger antioxidant than that of theaflavin. Moreover, the digallate derivatives of theaflavin demonstrate the increased antioxidant activity. Second one is allicin, diallyl thiosulfonate, is the biologically active compound mainly found in the garlic extracts. Allicin is known to possess various biological activities including the antibacterial, antifungal, and inhibition of cancer promotion. Moreover, allicin is known to reduce serum cholesterol and triglyceride levels, as well as atherosclerotic plaque formation and platelet aggregation. Until now, a variety of biological EECs of allicin were attributed to antioxidant activity. However, recently it has been found that the active ingredients responsible for the antioxidant property of garlic is 2-propene sulfenic acid and not the allicin. Thiosulfonates undergoes cope elimination to form sulfenic acids, thioaldehydes, or thioketones. The SS bond in the thiosulfonate is much weaker than the SC bond in a sulfoxide. Hence, this process can occur at room temperature. Cope elimination is even more susceptible for the allyl and benzyl thiosulfonates, such as allicin. Because of the weak CH bond of the allyl moiety, allicin is known to undergo cope elimination at room temperature to give 2-propene sulfenic acid and thioacrolein as shown in the figure. This figure demonstrates the mechanism of the radical scavenging activity of the allicin. The radical scavenging activity of allicin involves H-atom transfer to a peroxyl radical from the methylene of the allyl group on the divalent sulfur. This figure demonstrates an alternative mechanism, where the radical scavenging activity of allicin can be accounted for 2-propene sulfenic acid, which is produced from allicin by cope elimination. 2-propene sulfenic acid is reported to be over 1,000 times more reactive toward OOH* radicals than allicin. Third one is piperine, 1-piperoyl piperidine. Is an alkaloid present in fruits of black pepper, Piper nigrum, long pepper, Piper longum, and other piper species, family, Piperaceae. Piperine possesses many pharmacological activities, including anti-inflammatory, analgesic effect, anti-ulcer activities, antidepressant effect, cognitive enhancing effect, cytoprotective effect, and antioxidant activity. It is interesting to notice that the higher concentration of piperine results in the increased production of the OH*, whereas, in low concentrations piperine acts as an antioxidant. Piperine demonstrates synergistic antioxidant activity by doubling the absorption of dietary curcumin. Next example is curcumin, a lipid-soluble active principle of turmeric. Is a bis-alpha, beta unsaturated beta-dictone that exhibits keto-enol tautomerism. Curcumin shows remarkable antioxidant activity, and it has been found to be an excellent free radical scavenger. Curcumin has a chain-breaking antioxidant ability comparable to that of the vitamin E. As shown in the figure, the free radical scavenging activity of curcumin is correlated to the phenolic OH group and the CH2 group of the beta-dictone moiety. The free radical can undergo electron transfer or abstract H-atom from either of these two sites. However, pulse radiolysis and other biochemical methods credited the antioxidant activity of curcumin to its phenolic OH group. The figure depicts the mechanism for the autoxidation of curcumin initiated by hydrogen abstraction from one of the phenolic hydroxyl groups. The phenoxyl radical moves into the carbon chain, leaving a quinone method that is eventually quenched by the water molecules. The method radical performs a 5-exocyclization with the double bond to give the cyclopentadione ring and generating the carbon-centered radical. The reaction of curcumin with the molecular oxygen, O2, results in the peroxyl radical. The peroxyl radical is then reduced to the hydroperoxide by abstracting a hydrogen atom from another curcumin molecule, propagating the autoxidation chain reaction. Subsequently, the hydroperoxide loses water and rearranges into the spiroepoxide. The hydrolysis of the epoxide by the water-derived hydroxyl group results in the formation of the final bicyclopentadione product. It has been found that the copper complex of curcumin Cu, show promising SOD activity, with improved antioxidant efficacy. The mechanism of the O2* scavenging activity of the curcumin Cu complex is depicted in figure. When O2* are allowed react with the curcumin Cu complex, a major fraction of O2* reacts with Cu2 plus moiety, while only a small fraction reacts with curcumin. The reaction causes reduction of Cu2 plus to Cu plus. The Cu plus undergoes subsequent oxidation by another molecule of O2*, thereby regenerating the parent complex. Therefore, the catalytic activity comes mainly from the reversible redox reactions within the Cu2 plus or Cu plus couple in the complex. However, in presence of the excess O2*, the phenolic moiety undergoes oxidation resulting in the production of the phenoxyl radicals. Then these phenoxyl radicals can generate new products or react with reduced copper ions of the complex, resulting in the regeneration of the complex. In vitro methods for evaluation of antioxidant activity. Various in vitro methods are available for the evaluation of antioxidant activity of different compounds. Most important one is DPPH assay. that is assay of 1,1-diphenyl 2-picryl hydrazole, DPPH, free radical scavenging activity. DPPH is a stable free radical that can accept an electron or hydrogen radical to become a stable diamagnetic molecule. Due to its odd electron, the methanolic solution of DPPH shows a strong absorption band at 517 nanometer. As shown in the figure, the DPPH radical reacts with suitable reducing agent producing new bond, thus changing the color of solution. The solution loses color with the increase in the concentration of antioxidant as the electrons taken up by DPPH radical from the antioxidant. Such reactivity has been used to test the ability of compounds or plant extracts to act as free radical scavengers. Reduction of the DPPH radicals can be monitored spectrophotometrically by the decrease in absorbance at 517 nanometer. Let's see next topic: anticaking agents. They are food additives that keep powders or granulated materials such as milk powder, powdered sugar, tea and coffee powders, used in vending machines, table salt etc., flowing freely. Anticaking agents, in fact, prevent the formation of lumps, making these products manageable for packaging, transport, and for use by end consumer. Examples: vending machine powders, coffee, cocoa, soup, milk and cream powders, grated cheese, icing sugar, baking powder, cake mixes, instant soup powders, drinking chocolate, table salt. Types of anticaking agents: manufactured or man-made. Natural anticaking agents. First, let's see about manufactured or man-made anticaking agents. Most of the anticaking agents are made from synthetic substances such as silicon dioxide or magnesium and calcium stearates, solid saturated fatty acids. Some anticaking agents manufacturers also produce organic and hypoallergenic anticaking agents from natural source such as rice. Majority of anticaking agents are found with E numbers from 500 to 599. Examples of manufactured anticaking agents include calcium silicate, E552, magnesium carbonate, E504, and sodium aluminosilicate, E554, etc. Next type is natural anticaking agents. However, there are many anticaking agents that come from natural sources. Some of the natural anticaking agents include kaolin, E559, talc, E553b, and bentonite. Dual-purpose anticaking agent. However, apart from the list of E numbers of anticaking agents, there are a few that fall into other categories too, like the acidity regulators, because they serve two purposes. Some of such anticaking agents having dual or multiple purposes include E421, mannitol attacked as anticaking agent and also a texturizing agent, a sweetening agent, an anti-sticking agent, as well as a humectant. E460A, microcrystalline cellulose as anticaking agent, also a bulking agent, binder, and stabilizer. E460B, alpha cellulose as anticaking agent, also a bulking agent, binder, and stabilizer. Next topic: anti-foaming agent. The presence of foam can result in numerous troublesome issues, including the loss of target products and substrates, reduction in the equipment working volume, and environmental pollution. Let's see about formation of foam. In the processing of food products or beverages comprising proteins, there is a high tendency for foaming. Because of their amphiphilic structure, which gives them the flexibility to unfold and adsorb at the interface, protein molecules act as surfactants and create strong intermolecular interactions. This generates a viscoelastic, irreversibly adsorbed layer at the air-liquid surface, which stabilizes the foam. The foam is a nuisance as it results in a severe loss of production capacity. deterioration in the quality of foods as well as a loss in volume efficiency. Anti-foaming agents and techniques have therefore been introduced to counter this foaming process. Properties of anti-foaming agents: decrease the surface elasticity of liquids, thereby preventing the foam to attain a state of equilibrium between the surface elasticity and the anti-foaming agent. This will destabilize the foam and interfere with the foam formation process. Mechanisms of anti-foaming and foam destabilization: it acts as hydrophobic bridges between the respective phases of film surfaces, and thus discontinue the foam formation process. When a hydrophobic defoamer is at the interface between the two respective liquid phases, it triggers the formation of a convex surface in the foam itself. This convex film will cause the liquid phases to flow away from the anti-foamer, which now behaves as a hydrophobic bridge, because of the buildup under the convex curved surface, which automatically causes the destabilization and collapse of the foam. Another possible mechanism of foam destruction involves the displacement of the adsorbed protein molecules, which cause the foam generation on the foam film surface, thus interrupting the stabilizing action of the protein. This mechanism disturbs the equilibrium on the foam film, causing the destruction of the foam. Another mode of defoamers spreads rapidly over the surface of the foamer film to squeeze the liquid, thus destroying the foam. Natural defoamers: there are defoamers of natural origin such as the Quillia extracts. Quillia extracts can be found naturally by performing aqueous extraction of the bark Quillagia saponaria molina plants. Compounds obtained from aqueous extracts of Quillia include saponins, tannins, polyphenols, and calcium oxalate. Although polyphenols and tannins form the major components, Quillia extracts have also found use as foaming agents in soft drinks and other food items. Synthetic defoamers: the most commonly used types of defoamers in bioprocesses include those which are silicone-based, oil-based, water-based and silicone surfactants. Example: polydimethylsiloxane, PDMS, polyoxyethylene, PEO, and polyoxypropylene, PPO, glycol-based. Okay students, that's all about today's session. This session elaborates the mechanism of action of the natural antioxidant compounds, and assays for the evaluation of their antioxidant activities. Also about anti-caking and anti-foaming agents. Thank you students.